That is on page 52 of your textbook. Okay. This is the last lesson for this chapter 2. Okay. Today we're going to talk about enzymes. Okay. Vamos a hablar de lo que está en el LMS directamente. Ok. Está directamente ahí. No es la clase como tal. Lo envía luego. Ok. Okay. So we're going to start with enzymes. Okay. The key concept for this lesson is that enzymes are catalysts for chemical reactions in living things. Okay. Last lesson we were talking about chemical reactions. So when we talk about enzymes, are catalysts for chemical reactions. Okay. A catalyst is a substance that decreases the activation energy needed to start a chemical reaction. Okay. So when we talk about enzymes, are what are used for chemical reactions so we can have a lower activation energy. Okay, that's important. We said that the activation energy is the energy needed to start the chemical reaction. Okay, so a catalyst basically what it does, it lowers the activation energy, so we need lower energy. Okay, so that's what a catalyst is. Okay, so catalysts are substances that speed up chemical reactions. Okay, if we want a chemical reaction to occur more fast, we use what we call a catalyst. Okay? So enzymes are a type of catalyst that we use to speed up chemical reactions. Okay? So what does a catalyst does? Okay? It basically decrease the activation energy needed to start that chemical reaction. Okay? So that's what we call a catalyst. Okay? It also increases the reaction rate. So we're going to have a quicker reaction, okay, a quicker chemical reaction because it also increases the reaction rate. Okay, we can see in this diagram how this works. Okay, you see that an activation energy on catalyzed, that means it doesn't have the catalyst. What happens? You see it has a higher activation energy. What happens when we use a catalyst? Okay, the activation energy decrease, so we need lower energy or we need less energy to start the chemical reaction. ¿Lo puede ver? Okay, so we got in the first line, we got a normal reaction, and in the second line, that is under, ¿verdad? Con esos puntitos, we got a catalyzed reaction that needed less energy, okay, to start the chemical reaction. Okay, any questions about a catalyst? That's what basically a catalyst does. Okay, it decreases the activation energy and it decreases the reaction rate. Estamos, preguntas, dudas. No. Okay, perfecto. Let's continue. Tienen que ponerle mute al micrófono, a menos que haya una pregunta. Okay. So, le voy a pedir favor. Okay. So enzymes allow chemical reactions to occur under tightly controlled conditions. So also enzymes allow reactions to occur under controlled conditions. Okay, that's very important. Okay. Okay. So enzymes are catalysts in living things. Okay. Victoria, ¿se puede ponerle mute al micrófono? No problema. Okay. So enzymes are needed for almost all processes. Okay. So that's also very important. Okay. Also, we can say that most enzymes are proteins. Okay. We know how proteins are made. We saw that on the past lessons. Okay. Also, we can say disruptions in homeostasis. Who can tell me what is homeostasis? Eso lo hemos visto. Okay. ¿Verdad? Lo último que dijimos es que most enzymes are proteins. Okay. La mayoría de las enzimas son proteínas. Okay. Who can tell me where proteins come from? From one carbon-based molecule does proteins come from? Oh, ¿verdad? ¿De qué aminoácidos, perdón, de qué carbon-based molecule vienen las proteínas. Okay, proteins come from amino acids. Okay, that's from lesson 2.3. Okay, vamos a continuar. Okay. So a disruption in homeostasis can prevent enzymes from functioning. Okay, who can tell me, who remember what is homeostasis? ¿Qué homeostasis? Lo habíamos visto en el capítulo 1. Who can tell me very fast? ¿Alguien se acuerda? Exacto. Homeostasis is basically that, okay? It's the maintenance of conditions in the interior body, okay? En the interior of the organism, okay? Básicamente eso. So, a disruption in the homeostasis, que ese conditioning, can prevent enzymes from functioning, okay? So, it's obviously that enzymes or need a temperature. Okay, so enzymes function best in a small range of conditions. Okay, so when an enzyme is under bad conditions, okay, the enzyme doesn't gonna work or does not gonna work like it's supposed to work. Okay. For example, changes in temperature and pH, ¿verdad? El pH que habíamos visto from acid to base can break hydrogen bonds. Okay. So if hydrogen bonds are broken, what's gonna happen? Well, those functions gonna be broken down. Okay, so the enzymes doesn't gonna work like they're supposed to work. Okay, so in other words, if we have a catalyst, so a catalyst, it makes that the chemical reaction occur faster because it lower the activation energy needed for the chemical reaction to start. Okay, for example, if changes in temperature or pH break the hydrogen bonds, we can see that the enzyme function gonna be disrupted. Okay, so an enzyme function depends on its structure. Okay, así que una enzima, si una enzima, ¿verdad? Su estructura, ¿verdad? O su función se ve dañada, ¿qué va a pasar? Pues la estructura va a estar dañada. Okay, so an enzyme functions depends on its structure. Okay, duda pregunta de eso. Okay, we can see that on page 53 of our textbook. Okay, so enzymes are catalysts for chemical reactions in living things. Okay, any questions about this? Okay, so it's very important that we got the conditions required for the enzymes or catalysts to work very perfect. Okay, so enzyme functions best in a small range of conditions. Okay, preguntas de esto. Okay. An enzyme structures allow only certain reactants to bind to the enzyme. Okay. So let's see this. Okay. That's what we call substrates. Okay. Substrates are the reactants that an enzyme acts on are called substrate. Okay. That's where the enzyme gonna act on. Okay. And the place that it's gonna act is the active site, el sitio activo. Okay. Okay, this is what we are talking about. Okay, we got the enzymes. Okay, we got the catalyst. Okay, so the catalyst or the enzyme gonna go to this active site in the substrate. Okay, the substrate is the place that the enzyme gonna bind together. Okay, so when that happens, when the substrate interacts or the enzyme interacts with the active site, with the active site, what's gonna happen? That's gonna lower the activation energy needed for the chemical reaction to start. Okay, so the substrate is the place where the enzyme or where the substrate, gonna act. Okay, so the substrate bind to an enzyme at certain place. Okay, el sustrato, mi gente, que en otras palabras es el reactivo, es lo que va a interactuar con la enzima. Okay, so when we got the enzyme, it got a structure that allow only certain reactants to bind to the enzyme. Okay? cada enzima tiene su sustrato, ¿verdad? O cada enzima tiene su sustrato que va a interactuar con, ¿ok? Estamos. So those two parts, estas dos partes, ¿ok? The substrate gonna go to the active site. Usted ve que es como si fuera un rompecabezas. Eso va a interaccionar, ¿ok? It's gonna interact to the bind. It's gonna bind, the reaction is gonna bind to the enzyme, ¿ok? At certain places, ¿ok? Why certain places? Because it's like a key, ¿ok? Parece como una llavecita que va a interactuar con esa enzima, ¿ok? Lo notas, la parte amarilla o la parte mostaza is gonna interact with the top part, and the blue part is gonna interact with the lower part of the substrate, okay? And de la enzima, okay? Estamos. So that's basically what we call a substrate and an active site. Ese sitio activo, okay? Okay. So we got basically the lock and key model help us illustrate how enzymes function. Okay. So substrates are brought together. Okay. Bonds in substrate weaken. So basically, this is what we are talking about. Okay. We got the substrate bind to an enzyme at a certain place called active site. So those active sites, are where the substrate gonna bind together with the enzyme. Okay. So when this is all bind together, we got a figure like we see in the second diagram. Okay. The enzyme brings substrate together and weakens their bonds. Okay. So that's gonna happen. Okay. When the enzyme interacts with the substrate, the bonds of hydrogen, the hydrogen bonds are gonna be weakened. Okay. So the catalyzed in the third diagram, we see that the catalyzed reactions form a product that is released from the enzyme. Okay. After the interaction of the substrate with the enzyme, we're gonna see a product being released from the enzyme. Okay. ¿Qué habíamos dicho, estudiante? The enzyme is the catalyst. Okay. So the catalyst, what it's gonna do is gonna lower the activation energy needed to start the chemical reaction. So the product is gonna be the green thing, okay, that is binded together, okay. So that's what is released from the enzyme, okay. So a question we can answer is describe two functions of catalysts in chemical reaction. Who can tell me what are two functions of catalysts, in chemical reactions? Who can tell me two functions of catalysts in chemical reactions? That's a question we can make. ¿Quién me dice? Según lo que hemos hablado hasta ahora. What are the functions of catalysts? De la enzima, en otras palabras. Nadie sabe. Okay, that's the first function. Thank you, Adriana. The second function of the catalysts are that increase the reaction rate. Okay. En otras palabras, the catalyst decrease the activation energy, but increase the reaction rate. Va a incrementar cuánto, ¿verdad? Cuánto producto o cuánto, ¿verdad? Vamos a tener de ese producto. Okay. So those are basically the functions of a catalyst. Another question that we can make is how does the structure of an enzyme affect its function? Who can tell me? Again, how does the structure of an enzyme affect its function? Okay. ¿Cómo la estructura de una enzima se ve afectada por su función o afecta la función de la enzima? Eso lo dijimos hace unos segundos, okay? If the structure of an enzyme changes, its function will change also, okay? So basically, the enzyme, the structure of the enzyme is strictly related to the function, okay? If the enzyme structure change, the function of the enzyme will change, okay? So we don't want the structure of an enzyme to change because also the function will change, okay? So that's basically those two questions that you have to ask yourself, okay? Any question about this lesson, okay? This is basically the lesson 1.6, okay? Let me see if something else is on the presentation. Okay, okay, so this is basically the lesson for today, okay?